we gonna do in this video, brain? The same thing we do in every video, Becky. Try and take over the world with science!
Hello everyone, my name's Rebecca and I'm a neuroscientist here to talk to you today about all things neuro and science. And in this video I wanted to talk about one of the best questions I think I've actually been asked while I have been out and about doing science communication. I do a lot of talking to a lot of different groups of people. It's actually getting me to shut up that's the trick, and no one's quite managed that yet. But while I was out doing this particular talk, I had a gentleman come up to me and ask, so if one part of the brain does one thing, then why is it so difficult to come up with drugs that fix neurological conditions? I mean, if you know what part of the brain is going wrong, why can't you just hit it with a drug and fix it straight away? We know that memory is associated with the hippocampus, so fix the hippocampus and get rid of Alzheimer's disease. We know that movement is associated with the motor cortex, so fix the motor cortex and fix movement-based disorders like Parkinson's disease. In this video, I wanted to address why making drugs for the brain is so tricky. But first, let's start out with addressing why that question is actually a really good one. And that's mainly because it's based on a really solid intuition that we call rational drug discovery. See, the idea of figuring out what's wrong with the brain and then finding a drug to fix that is actually what we've been trying to do for the last few decades. But at one point, this was a groundbreaking idea. Gertrude Elion, George Hitchings, and James Black won a Nobel in physiology and medicine for their systematic approach to designing drugs, which focused on first understanding the biological mechanisms which underpinned a disease. And this led to new drugs for leukemia, malaria, and much more. But you might be thinking, if that won a Nobel in 1988, what the hell were we trying to do before that? And the simple answer is basically trial and error. We would screen huge numbers of compounds looking for potential hits, i.e. things that could maybe be drugs later on. And in the cases of many mental health conditions, the drugs we still use today were actually originally used for other purposes, as sedatives or anti-epileptic medications. For example, methylphenidate, often better known as Ritalin, is now a commonly used medication to manage ADHD, but was originally intended to treat chronic fatigue and narcolepsy before doctors started to recognize its other benefits. Essentially, drug discovery was a throw compounds at machine or throw drugs at patients and see what sticks approach. So, the idea of rational drug discovery is a good one, and one that has been effective outside of neuroscience. Find biochemical root of problem, hit with drug, improve illness. So in answer to the question, why don't we just find the problem in the brain and then target it with a drug to improve neurological conditions? The answer is, that is kind of exactly what we're trying to do. But unfortunately, as with many things in science, it's just a touch more complicated than that. And so I'm going to go all Jenny Nicholson on this and address those complications in a numbered list. Complication 1. One brain area doesn't actually do one thing. In my very first video, I introduced the idea of source localization, that certain parts of the brain have certain jobs to do. And whilst this is broadly true, even in that initial video, I acknowledge that there are limitations to this approach. For example, neuroplasticity, the brain's ability to just rewire itself. Imagine making a map of a country, but new railways and roads are constantly springing up, which means there can be mass migration at a moment's notice, and suddenly London is a couple hundred miles north of where it used to be. Also, even though London is largely known for exporting little red buses and small snow globes of Big Ben, when there's a sudden shortage of fake snow in the south, Liverpool starts making the snow globes instead. Great, just to confuse us. Also, London actually gets its red paint for those little buses from Cornwall, which means if Cornwall gets flooded, London is going to stop making those little red buses, even though there's nothing actually wrong with London. And Cornwall gets the dye to make the red paint from the Moulin Rouge in Paris, so suddenly a problem in Paris is causing a lack of red buses in London, and all we can see is there's no more little red buses. And so we might assume that's because of a London problem, but it might actually be because of a problem miles away, and whoop, London moved again. Starting to see the problem? The brain is based on a whole lot of underlying circuitry. Brain areas are heavily interconnected, which means even functions we've studied a lot, like memory, still can't be localized to one brain region. One region does not equal one job, and one job does not equal one region. And to add one final complication, another lovely gentleman once asked me, so what percentage of the brain do we actually understand? A great question, and to answer that, let's go over to Rebecca on the weather. Thanks, Rebecca. Returning to our map, we know this much of our map. The known known. There are also some lands people have seen on the distant horizons that haven't been mapped out yet. The known unknowns. But without any more information than that, how can you estimate what percentage of the world we understand? Short answer, you can't. Because there are also so many unknown unknowns, things we don't even know we don't know yet. And without knowing the unknown unknowns, it's impossible to even guess at how much of the map we've truly seen. Despite the textbooks giving nice, neat answers where memory is located here and language is located here, the truth is that we're still learning a lot about the brain and where those functions lie. And this is made more complex by the fact that there's a lot of unknown unknowns and also a lot of circuitry linking all of these regions together. And this makes it very difficult for us to tell exactly what brain region is responsible for certain symptoms in disease. This also ties into complication number 2. Neuroscience is a wee baby. The forefathers of modern neuroscience have been around for centuries, from phrenology to psychology, but it wasn't until 1971 that we could actually see the living human brain for the first time using a CT scan. That's just over 50 years ago. And it wasn't until the 90s that we got our first functional brain scanners, which actually allow us to look at changes in neural activity in real time. Before these scanners, we relied on post-mortem studies, looking at the brain after death. And while these studies are still incredibly helpful even today, there's only so much you can tell about a brain when it's not working anymore. It's like looking at a computer when it's been turned off. Sure, you can still tell a lot about it from the hardware, but you can't actually see it running. Animal studies were also the bread and butter of neuroscience and are still incredibly valuable today, but I don't know if you know this, it's quite a technical point, so you'd be forgiven for not coming across it before, but mice? Not people. Yeah, and their brains do look quite different. They're smooth. Animals also don't experience some symptoms in the same way as humans, which can make it difficult to create animal models of mental health conditions like depression, for example. Now, this is a problem even today, despite the fact we have all our lovely brain scanners, because despite the fact they can be controversial at times, animal models are a key way that we deal with complication 3. What's the mechanism? Hit brain area with drug sounds good, but what exactly will the drug do becomes the next question. In my video on dementia treatments, I cover some of the ways in which we can use drugs to alter processing in the brain, with perhaps one of the most common being to increase or decrease the level of certain chemicals. But this approach doesn't work for everything, and we need to know exactly what chemical we'd like to hit and how in order to have that positive impact. It goes back to our idea of rational drug discovery. It's not enough just to know what brain region you want to target, you want to know how you're going to tweak it in order to fix a specific issue. This is why trial and error isn't actually a bad strategy for drug discovery, because we get to have all these positive effects of drugs without having to understand the exact biochemical cause of each illness, which, as we now know, is no mean feat. Take depression. Antidepressants actually came about pretty much by fluke before the idea that depression was caused by a loss of serotonin. In fact, the idea that depression is caused by a serotonin deficiency has been in large part supported by the fact that antidepressants, which act on serotonin, have been so effective. Treatment came before cause. Serotonin is also the molecule on my necklace, just for anyone curious. But fun fact, this kind of reasoning is actually a logical fallacy called the treatment etiological fallacy. Essentially, a symptom isn't necessarily caused by the same mechanism that we can use to treat it. For example, headaches are probably not caused by a lack of paracetamol, and yet taking paracetamol will improve your headache. In a similar way, though we know that antidepressants can be super effective for a lot of people, we are now vastly re-evaluating the idea that depression is actually caused by a loss of serotonin. So rational drug discovery is great in theory, it's sensible, but it can also be incredibly difficult to find the biological cause of an illness and by extension a druggable mechanism, especially in neurology and psychiatry. But let's say for argument that you have found a druggable mechanism, you know what part of the brain you want to target and how. Let's even ignore all the non-scientific factors that can slow down drug discovery, like money and paperwork. Even then, there's still one more barrier in your way. Literally. Complication 4, the blood-brain barrier. See, your brain isn't as easy to reach as the rest of your body. It's actually sealed off by the blood-brain barrier, which is made up of highly selective cells that only allow VIMs through. It's very important molecules. I make myself laugh. These cells care not for your attempt to make a helpful drug. 98% of small molecules and 100% of large molecules will ping off the blood-brain barrier. And guess what we usually use to make drugs? It's them, yeah. This makes drug design even trickier for drugs that are trying to target the central nervous system and heavily restricts what we can use. It means we have to get real creative using nanoparticles and transporters to smuggle our molecules across the blood-brain barrier. And I think that's actually all the complications I can think of for now, so other than all that, it's easy. So, why is it so difficult to make drugs that fix the brain? Firstly, because the brain is so heckin' complicated, compounded by the fact that we haven't been able to study the brain in a living human for all that long. And even once you have those druggable mechanisms and those potential causes, there's still extra steps in your way, like that pesky but helpful blood-brain barrier. But even with all that, we're still seeing some incredible advances in drug discovery at the moment, with new approaches, new mechanisms, and fingers crossed, some new hope. That's all the time that we have for today, but if you have any questions about the neuroscience in this video or suggestions for future videos, please do leave a comment down below. Other than that, it's time for me to head back to the lab and plan what I'm going to do for the next video. But until then, please do let me know of any progress you make in trying to take over the world with science!